love that we can use the language of physics to talk about something like cancer.
Yeah. I mean, physics is everywhere. We've heard about aneuploidy. That's how you get Down syndrome, right? You get 3 copies of the chromosome 21. We should only have 2 copies. Aneuploidy is when we have more or less of a specific chromosome. It is very bad and catastrophic for normal cells. Cancer cells love it. Okay. 90% of solid tumors are aneuploid tumors.
Okay.
Here you can look at a cancer cell. There's 4 of 2, 4 copies of 4, 3 copies of 5. If somebody looked at this chromosome, it's like there's no way this is a normal cell.
Right.
Because there's what is going on? If we were to look at all of the sets of all the different chromosomes that we'd have to model some kind of simulation of cancer evolution, it would be something like 10 to the 10 to the 20. It's a lot. It's never going to happen.
You're right.
So instead, these researchers came up with AlphaK, which is inferring what's called a local landscape of fitness. And it's trying to figure out how the tumor is going through this local landscape. Let's talk about something called the fitness landscape, a 2D landscape that has hills and valleys. Okay. The direction north-south could be how much of gene A do I have? East-west could be how much of gene B do I have, okay? So every point is a location in genetic space. There would be hills, which is where you're more fit, so it's an advantage to be here. And there's valleys where there's less fit, right? Now, this is a nice metaphor, and for the longest time, it was just a metaphor. What these guys have done with the AlphaK concept is operationalize that metaphor into something that's quantifiable. It's taking that metaphor and it's saying, actually, this is a real thing that we can at least apply very quantitatively to the problem of aneuploidy. Because in aneuploidy, your axes don't have to be genes. They can be number of a certain chromosome. You can have this axis be, I have 1 of chromosome 1. I have 2 of chromosome 1. 3, 4, 5 copies. The other axis will be, how many of chromosome 2 do I have? How many of chromosome 3 do I have? The 4th axis. And so you have 22 dimensions, each chromosome. So now we can create an actual quantitative landscape. And we can measure at each point, what is the slope of this landscape? Because we can track that cell at that particular point and ask which way did it go?
What we're saying is now scientists are actually taking real world data and creating a literal fitness landscape.
Yeah.
Particularly in oncological studies.
Yes. And then from that construction, now we can tell which way is it going to go. So with this paper, we can actually predict cancer drug resistance before the fact, before it actually happens, right? And then we can be like, hey, maybe we shouldn't use that one.